disease and to produce or elicit a rapid and protective immune response that would shorten the disease time and lessens morbidity and mortality. The treatment consists of inhalation of biodegradable liposomes called alpha-gal sialic acid liposomes. It's in early stages of the it's in early stages of the research, and since I recently retired from UMass Medical School, I'm looking for collaborations, maybe in this audience, that would be interested in advancing this research, and if it's found effective and safe, bringing it up to clinical trials. The alpha-gal sialic acid liposomes are liposomes that present multiple carbohydrate epitopes of two types. One of them is called the alpha-gal epitope, where you have a galactose-linked alpha-1,3 to a penultimate galactose on carbohydrate chain of glycolipids, and the other is sialic acid epitope, where sialic acid, terminal sialic acid, is linked alpha-2,6 or alpha-2,3 to the penultimate galactose on a carbohydrate chain similar to that of the alpha-gal epitope. The therapy is based on two well-established phenomena. One of them is well known to the audience in this room. This is the ability of the flu virus to bind to sialic acid epitopes. And the other is the binding of the natural anti-gal antibody to these alpha-gal epitopes. Anti-gal is the most abundant antibody in humans. It's 1% of immunoglobulins in every one of us. And in fact, this is the only natural antibody that we know that everybody has it without any exception, unless you are severely immunocompromised. The antibody was found in my lab some 32 years ago, and for quite a number of years, I've been interested in developing methods for exploiting this antibody in clinical settings. Now, the alpha-gal epitopes is produced as millions of epitopes per cell in all non-primate mammals, whether they are marsupials or placentals, and in part of the primates, prosimians like lemurs that evolved in Madagascar, and New World monkeys those are monkeys of South America. In all these species, the enzyme, glycosylation enzyme called alpha-1,3 galactosydeotransferase, link the terminal galactose to the penultimate galactose, as you can see here. And all these species don't have the anti-gal antibody. In contrast, old wood monkeys, those are monkeys of Asian, Africa, apes, and humans, they all produce very large amounts of the alpha-gal epitopes, but they completely lack the huge amounts of the anti-gal antibody, but they completely lack the alpha-gal epitope. We found that the reason is that approximately 20 million years ago, the gene that encodes for the alpha-1,3 galactosydeotransferase, this gene was inactivated in this group. So they end up with the antibody, but no alpha-gal epitope. You might have heard about this antibody because it is responsible for the rapid rejection of organs from pigs, for example, xenografts that are transplanted into this group because of this anti-gal alpha-galactosyde epitope interaction. I will just mention that this antibody also appears in chickens and other birds since they don't have the alpha-gal epitope, and the significance of it I'll return at the end of my discussion. Now, since the anti-gal antibody, every one of us has it in large amounts, it can be exploited in the clinic. And the two mechanisms by which it can be exploited, one of them is when it binds to the alpha-gal epitopes, it activates the complement system, generates complement cleavage chemotactic peptides like C5a and C3a that generate a chemotactic gradient, inducing the migration of macrophages, as you can see here in the famous picture from National Geographic years ago. Once the macrophage gets to the bacteria or any cell or particle coated with anti-Gal, it binds through its Fc receptors to the Fc tail of the antibody that coats these particles expressing alpha-Gal epitopes. This activates those macrophages, and in the zipper-like motion, they use the Fc receptor to bind to additional tails of anti-Gal and inducing phagocytosis of those particles. Now, we can exploit these functions of anti-Gal by using alpha-Gal sialic acid liposomes, which you can see here. They are made of phospholipids, and within there are anchored glycolipids with alpha-Gal epitopes and or with sialic acid. One can generate those liposomes by mixing in an organic solvent phospholipids, alpha-Gal glycolipid, and sialic acid glycolipids, and drying them, adding saline, using sonication to generate those liposomes. Those liposomes can be administered to patients in form of aerosol by inhalation. Consider a situation where a flu patient inhales such liposomes, which are illustrated here with the alpha-Gal epitopes and with the sialic acid epitopes. Those liposomes would land in the mucus that is lining the respiratory epithelium and start binding via the sialic acid epitopes, binding the flu virus in that mucus. This interaction, as you know, mediated by the hemagglutinin interaction with the sialic acid. Concomitantly with that, the anti-Gal antibody will bind to the alpha-Gal epitopes and activate the complement system, generate chemotaxis of macrophages that would migrate toward those liposomes and interact with the Fc receptor binding to the Fc portion of anti-Gal on those liposomes and induce internalization of phagocytosis of these liposomes and the attached virus, as you can see here. These macrophages at this point would start acting as antigen-presenting cells. They would destroy the virus and process the immunogenic peptides of the virus, present them on MHC molecules, actively transport the internalized antigens to the regional lymph nodes, where they will activate virus-specific helper T cells that will have B cells to produce neutralizing antibody against the virus, and cytotoxic T cells that would kill respiratory epithelium cells that are infected with the virus. I would show you later how this critical stage of internalization of the virus into those macrophages results in increase of approximately 100-fold in the immunogenicity of the infecting virus. We have so far generated liposomes that have the alpha-Gal epitopes on them. Within a month, I hope to be able to introduce to this also sialic acid glycolipid to generate the sialic acid, the alpha-Gal sialic acid liposomes. So we have done studies with these liposomes in order to demonstrate the ability of anti-Gal binding to the liposome to induce recruitment of macrophages. And in addition, we've done studies showing the ability of the anti-Gal antibody to target the flu virus to macrophages and other antigen-presenting cells, and by this increase the immunogenicity approximately 100-fold, as I said. The experimental model that we use was a alpha-1 galactosyl transferase knockout mice. Unlike normal mice, this is the only strain that lacks alpha-Gal epitopes and can produce the anti-Gal antibody. First study was we checked whether introduction of this alpha-Gal liposomes subcutaneously in a small sponge disc made of a biologically inert material, a polyvinyl alcohol. We inserted it and checked the migration or recruitment of macrophages to these sponges that contain liposomes, compared them to sponges that had just the excipient saline. you see that already after 3 days, you get infiltration of cells with morphology of macrophages that internalize large numbers of the alpha-gal liposomes. The number of these liposomes increased after 6 and 9 days to 400,000 and 600,000 per volume of 0.1 milliliters. These cells that were recruited did not appear, or there was only marginal migration to sponge discs that were contained saline rather than the alpha-gal liposomes. When we check these cells for their characteristic, what kind of cell type, by flow cytometry, they bound macrophage-specific antibodies for CD14 and CD11b, but we didn't see any CD4 or CD8 T cells, or we didn't see B cells as well in the recruited cells. Now, when you are introducing directly into the skin intradermal injection of these alpha-gal liposomes, you see they are here in this open area, within 24 hours you get extensive migration of macrophages to this area. They are characterized by binding macrophage-specific antibody called F480 linked to peroxidase. And these cells will be in this area for 2.5 weeks. Then they will disappear without affecting at all the structure of the skin. So you see that when you are administering liposomes expressing alpha-gal epitopes, they bind the antibody, generate chemotactic factors, and recruit macrophages. And it is reasonable to assume that a similar thing may happen when the liposomes will have both alpha-gal epitopes and the sialic acid epitope. Now, when these macrophages reach the alpha-gal liposomes, they bind to the Fc portion of the antibody on these liposomes via the interaction between this Fc tail and Fc receptor, resulting, as I told you before, in the phagocytosis of the virus and the liposomes. Here you can see scanning electron microscopy of these alpha-gal liposomes coated with the anti-gal antibody and incubated with 2 macrophages. You see in the inset magnification of this area. And those liposomes are in the size of approximately 200 nanometers. There are even smaller ones with the size of 20 or 10 nanometers. When they don't have the antibody on their surface, they don't bind to the macrophage because there is no this activity, this interaction. So now the question is how one can demonstrate the increase in the immunogenicity of the virus as a result of targeting to macrophages. As I said, we don't have yet both sialic acid and alpha-gal epitopes on the nanoparticles, but we could use directly the virus itself where we express the alpha-gal epitope on it. Here we took flu virus PR8, and in vitro we engineered it to express alpha-gal epitopes by incubating with recombinant alpha-galactosidase. And we assume that when you will inject it, and the virus is inactivated, used as a vaccine, when you will inject it, it would bind the anti-gal antibody that would further bind through the Fc portion to Fc receptor, induce internalization into the macrophages, and the macrophage will process the antigen, take it to the draining lymph node, express the peptides to T cells, elicit an effective immune response. So here you see we took those knockout mice and we immunized them either with the PR8 virus that was engineered to express alpha-gal epitope, or the parental PR8 virus that was generated in embryonated eggs. The mice received 2 1-microgram injections of the virus with the Ribi adjuvant. We tested the antibodies produced against the virus by ELISA 2 weeks after the second immunization. And you see, with the mice that were immunized with the virus expressing alpha-gal epitope, then you get approximately 100-fold higher titer of antivirus antibodies in ELISA than in the mice that were immunized with the parental virus, lacking the alpha-gal epitopes. Similar difference we saw not only in the serum, this is for IgG, but we saw also for IgG antivirus in lavage of the lungs of these mice. Moreover, when we check for IgA antivirus, this group didn't have any IgA antivirus generated in the lung lavage, whereas this group had a significant amount of this antibody. Question is whether this immunization has anything to do with increased protection against the virus. So we took 25 mice that were immunized with the virus expressing alpha-gal epitopes and 25 mice that were immunized with the parental virus lacking alpha-gal epitopes. We used them as challenge intranasal with 2,000 plaque-forming units, and we followed the mice for a month and a half. Here are the data for the first 2 weeks. Afterwards, they remained the same. You see that in the group that was immunized with the virus expressing alpha-gal epitopes, only 3 mice died and 22 of them survived. Whereas in the group that had the immunization with the parental virus without the alpha-gal epitope, 3 of the mice survived and 22 of them died. So you see that this targeting of the vaccinating virus to the macrophages by this anti-gal antibody can increase very markedly the immunogenicity of the virus. Now, this can be achieved rather simply when you use vaccines of the virus. You don't have even to use the intervention of the recombinant enzyme. What one needs to do is to propagate the vaccinating virus in cell lines like vero cells, however, that are engineered to contain large amounts of the alpha-1,3-galactosyl-transferase enzyme in order to generate these alpha-gal epitopes, which you would end up with around 3,000 such epitopes per virus. But this method is not, of course, practical in the case of patients, symptomatic patients that have the virus infection. And for these patients, one can use these alpha-gal sialic acid liposomes because instead of the alpha-gal epitope being on the virus itself, it would be on the liposomes that the virus is attached to it. And therefore, the targeting of the virus would be at that time via the liposomes, as we can see here, and there shouldn't be reasonable, it's reasonable to assume that there shouldn't be differences in the increased immunogenicity as long as the virus is targeted to the macrophages. So just to summarize, I propose that inhalation of alpha-gal sialic acid liposomes will result in their landing in the mucus and functioning as a decoy binding the virus via the hemagglutinin sialic acid epitope interaction. Concomitant binding of the anti-gal antibody would generate chemotactic factors that would recruit the macrophages. Macrophages reaching those liposomes will internalize the liposomes and the attached virus, turn into antigen-presenting cells that induce an extensive immune response by fast propagation, fast processing of the peptides, and transporting of the internalized virus antigens to the to the draining lymph nodes. So this altogether would result in faster immune response that would shorten the disease and thereby will prevent future long-term complications. Just one, one sentence more. I mentioned to you before that chickens also express the anti-gal antibody. So it is possible that the mechanism that we hypothesize here would increase the immunogenicity against virus infecting humans may do it also in chickens. So you can envision the situation that if it's effective, you can take a flock of chicken which is suspected for infection by the virus, put it in a closed space and spray the area, introduce aerosol that of alpha-gal sialic acid liposomes that will be inhaled by the chicken, and it will be of interest to see what's the efficacy in stopping the infection. I will end up with the names of the researchers that collaborated with me helping in this project, and I'll be glad to answer questions, and later on, if any of you is interested in further collaboration, I'll be glad to discuss with you. Thank you.
Thank you for the great talks. Now this paper is open for, yeah, go ahead. Oh, the
I was just curious. I think it was a great talk. Thank you very much.
Thanks.
You've done the mouse work now. We know mice lie. Basically, it's a standard thing. Have you moved into the ferrets?
No. And you can't do it in ferrets because
the lack of
they have the alpha-gal epitopes. Essentially, the only non-primate mammal that you can do it is either those
or medical
mice,
students.
or no, I say non-primate mammal. This, or you have knockout pigs also for that, or monkeys.
Yeah, absolutely. So you can't go into ferrets for this.
No.
Okay. Fine.
Unless someone knocks out the gene out of them.
Can you cover the other virus as well, or not the?
Yeah, theoretically, you can use, instead of sialic acid, any docking receptor on those liposomes based on the virus, the respiratory virus that you are looking into. So it could be either sialic acid, or it could be other carbohydrate, or maybe other type of docking receptors. This is all theoretical.
I'm just curious because it's targeted macrophage, and it enhanced the production of antibody.
Yeah.
And at the same time, it suppressed the immune responses, but it also affects the beneficial for the survivor.
No, I didn't say that it suppresses the immune system. It activates it increases the production of antibodies, as I said, around 100-fold. I didn't have time, but I can show whoever is interested later data showing that T cells specific for the virus antigens, whether they're CD8 or CD4, also they are increasing in the spleens of those mice in the order of between 10-fold and 100-fold.
Oh, you're right.
Okay. Yeah.
Okay. Are there any other questions? Go ahead.
That was my question. Hello. Yeah, I'm wondering whether
could you speak up a little bit, please?
Where you have a protocol, you ask for some collaboration. Whether you have a protocol in order to replicate your studies in another region of America, for example, Costa Rica or Central America.
Yeah, I can provide a protocol. Yeah, and one needs the the liposomes, which I can provide, as well as I can work with my former university to provide the mice which are needed for
so
that.
We can talk this.
So those are things that can be discussed. Yeah.
Okay, thanks.
Yeah.
On the back.
I have a question regarding lung damage. Do you see increased neutrophil activation in your model? Do you see increased lung damage due to increased macrophage activation with alpha-gal?
I don't know. I think that theoretically, if you will use high dose, which I don't know what it is compared to the optimal dose, you may reach also some kind of effects that are associated with large amounts of macrophages that may not be healthy, or neutrophils. So it's a matter of dosing. So those are issues that have to be studied experimentally and identify if there is an effective dose that is minimally damaging, identify it in the mice, and afterwards it will have to be done in a primate model, I would imagine, before going on to humans.
And